Hello students, here we are at Module 15, which means we just finished Module 14. So what we need to do is get a quiz on that one. So I believe that I put a quiz at Google Classroom and it's posted there and you can take it from there. Is that right? If not, I will just email it to you, okay? And if I email it to you, you could either type right on it and then save it and send it back to me, or you could print it out, write on it, take a picture of it, and send that to me. Sorry for all the trouble. I know that's a lot of steps there, but that's what we're going to have to do for now. Okay, so we are looking at botany, and we're going to be looking at plant reproduction here. So for this first part in 15A, we're actually going to be doing the kind of the middle part of the lecture right here. Okay, so on Lab 28 in your book, you're going to see a bunch of stuff there at the beginning, and that's not what we're doing today. We're going to do that next time. Although you will see a picture of a flower you're going to label in a place right there somewhere, right? Okay, so there's quite a bit of stuff today. So you see all that stuff about water processing and everything on Lab 28. That'll be next week's lecture, okay? So this week we're just going to concentrate on the flowers, and so you can kind of, for now, skip that beginning part. And on my book I can't really tell where it starts, but there's a place to draw a flower. Okay, so go to that spot in your book. So for the most part today you're going to be listening to this lecture. In your textbook we're starting at page 472, and that's like smack in the middle of module 15. We'll move through there. Just do the flowers to the flower dissection and the embryo sac formation, this kind of thing. And then we'll finish off the seeds and everything next week. So next week we'll talk about water processing in plants and then move to seeds and fruit as well. Okay, I hope that's not too confusing. So we're looking at insectivorous plants. Remember, plants make their own food, right, through photosynthesis. We know that. But in some cases we have plants who need a little extra help. They might be in some nutrient poor soil, or in a place where they're not getting a lot of sunlight, and therefore they have adapted to have these other strategies for getting nutrients, and that is to become insectivorous. In other words, they eat insects. All right, and the insects will give them vitamins. If they have enough sun, they can do photosynthesis, and they don't absolutely positively need to eat these. But like I said, sometimes they're not getting enough sun, and they have adapted this way to get this nutrient elsewhere. We're going to be looking here at a short video on the Venus flytrap, which is probably the most famous of the insectivorous plants out there. You're going to see there's a sensitive trigger. Once it is triggered, that movement will cause motion on the plant, and it will begin to digest the creature that has sadly walked into its path. So let's take a look at that video.
Note the Venus flytrap. Did you ever imagine that a plant could devour such large creatures?
All right, so then the plant will start to secrete these enzymes that will break down that poor creature, that poor little frog, right? Right here you can see a pitcher plant. This is where creatures will fall down into a deep chasm in there that has some digestive juices in there, and they'll start to be dissolved by it. Bugs and frogs as well. So that's one other way of getting some nutrients to plants. So we're looking at reproduction, and so all of module 15 is really about like how the water is processed, and then reproduction, and then the reproductive parts of the plants. So there is asexual reproduction and sexual reproduction in plants. So we'll be looking specifically at several of the vegetative reproduction strategies. That would be asexual reproduction, including elongation of stems, elongation of roots, plantlets that form, runners that form, tubers, and grafting. We'll be looking at those today. And then we'll spend a lot of time on sexual reproduction, which of course is the formation of flowers. Oh, I'm sorry, let me go back here. This right here is showing runners down here. So runners we have a original plant, right? It will send out a little runner, sort of like a little bud that comes off of there, a little extra stem action, and it'll actually rest on the ground and produce some roots and make another plant right here. Famous for this would be strawberries. Strawberries are runner plants, and so if you plant some of those, then they will, just stretch out with these new little runners coming off there. Also, if you happen to have St. Augustine grass in your yard, it also sends out runners, and you may have noticed this if you're trying to pull some weeds. Sometimes you'll pull up some of the St. Augustine, and a big old line will rip up from there. That's because it sent out all of these runners. Okay, we're going to watch a little video here, and I apologize for this video. The guy's voice is very annoying, but the information that he has is very good. Okay, I have looked for new videos that would take its place, and I just can't find anything that doesn't have its own problems as well. So just stick with me on it and listen to the content.
Vegetative reproduction.
Yeah, sorry.
Vegetative propagation is routinely done and is preferred by many plants. Some of the flowering plants propagate by specialized structures. This method of reproduction is asexual reproduction, where there is no involvement of male and female gametes. This method of propagation in plants is called vegetative propagation or vegetative reproduction. A large number of horticultural crops, such as guava, grapes, pomegranate, orange, and citrus, vegetative crops such as potato and sweet potato, and flowering plants like chrysanthemum are produced through vegetative propagation. Advantages of vegetative propagation. Vegetative propagation helps to maintain fixed qualities of the parent plant. Vegetative reproduction offers a uniform root stock for budding or grafting. This root stock can be selected from a plant that is immune to diseases. Different types of buds in plants. Stems usually grow above the soil and produce buds, leaves, flowers, and fruits. The place where a leaf rises on the stem is the node, and the portion between the two nodes is called an internode. The stem ends in a bud known as the terminal or apical bud. Buds are also present in the axil of leaves, known as axillary buds. Buds which contribute for growth are called vegetative buds, and those that give rise to flowers are known as flower buds. Buds can also arise on the roots or leaves. Nodes. Grass plants have a weak stem. The internodes are long, and the nodal portions will have roots and axillary buds. When they are cut at internodes, these buds develop into independent new plants. Eyes of potato. Potato tuber is a modified stem. The shape of these structures is similar to an eye. These are called the eyes of the potato. These eyes are cut from the tuber and germinated in the soil. Leaf buds. In Birophyllum, buds are situated in the depression of leaf margins. The usual position of buds is either at the terminal part of the shoot or in the axil of a leaf. These buds in Birophyllum are known as apophyllous. Root buds. In curry leaf plant and neem, these young plants develop from the buds present on the root. They develop into independent plants.
Okay, so you see there we have all those different ways of having vegetative reproduction, which does, like the video said, it helps preserve that, genetic strength, the genetic strengths. We're going to see in a minute, though, when we talk about sexual reproduction, how we have that genetic variation that comes in, which is also can select, if you will, strong traits. Now, plantlets are these little leaflets right here. You can see them growing on the edge of the leaf. These are like succulent plants. And that's just a little plant right there, right? And it can drop off and become its brand new plant. So that's vegetative reproduction. It didn't require any pollen, any pollination, anything like that. Over here I like this because you can see the little root parts coming off of that. So when that breaks off, it'll just plant those roots in the ground and make a new plant. Grafting is very commonly used. Specifically, it's used for fruit trees. So if you happen to have a fruit tree outside, the one that you've planted on your own, not a wild one, go out and check the trunk of it and see if you see a lump kind of low down on the trunk. Probably you will, because most fruit trees have gone through this system known as grafting. So grafting is when you take the stem of a plant that grows really great fruit and put it onto the root of a plant that is very hardy, that is very successful in its ability to absorb the water from the ground. You can see right here how the stem has been cut at this angle, and you slip it into, that's called the scion. The scion then slipped into the stalk or the root stalk, and then it's tied up like this. It will grow like a scar over it right here. They'll end up taking that tape off. When I say they, these would be the people who are growing fruit trees and get them ready for you. You don't typically grow fruit trees from a seed. You go buy a tree, right, that's already ready to go. And that's because they have made it better by putting it on a strong rootstock. There's a great story about this. The fruit that in France, they grow a lot of grapes because the French really love their wine. And so they have very old and highly regarded roots, stalks, and plants that they're growing in France to make wine. In the 1800s, something happened. There was a disease that came in when somebody actually brought in some plants from overseas. And these plants brought in a little bug with them called phylloxera. And phylloxera is a type of nematode, and it was down and it started to eat the roots of the native French vines, and they were ruining them. These 100-year-old vineyards were losing their grapevines, and it was tragic. You know, the French love their wine, so they had to do something, and quickly. So they started looking for a rootstock that was immune to the eating by these little phylloxera. They looked all over the place, and they found rootstocks that were successful. They brought the rootstocks in, and they grafted all of their French vines to this new rootstock that they found that was immune to the phylloxera. And it's interesting because the rootstocks they found actually came from Texas. So the reason that the Texas stalks were so hardy was because the Spaniards had brought those in. Sorry about that. Had brought those in to their churches that they planted way back in the 1500s. So that would have been the Alamo and places like that. You know, the priests needed to have their holy wine, and so they brought grape leaves with them, grape plants with them, and planted those. So in Texas, they had to be very hardy because it's dry, it's hot, and they became very resistant to any kind of disease. And so then in the 1800s, they took a bunch of the rootstocks from Texas over to France. They grafted them into the French scions, and to this day, most of the French grapes are growing in Texas rootstocks. How about that? Okay, that brings us now to the sexual reproduction of plants. This is kind of the highlight of the module, in my opinion, because we're looking at flowers, and flowers are just a lot of fun to study. So we're going to be looking at the components of sexual reproduction in plants, and those would be how the pollen is formed, how the embryo sac is formed, and then how pollination and fertilization take place in those places. We'll be looking at perfect flowers and imperfect flowers. If we had our lab today, we would be doing a dissection of a perfect flower. So what is a perfect flower? So this is a flower that has all the parts. It has the male parts and the female parts of the flower ready to go. They have both the carpel and the stamen. So we're getting to the part in your book where you're going to end up drawing this flower and understanding what the carpel and stamen are. So you can get a better idea of that. Here's a sample of a perfect flower. It's got the male parts right here, and then the female part in the middle. It's hard to see it in that picture. We see one here in this composite flower. And then, of course, our famous bluebonnet right here. Okay, so it has the parts it needs right in there. I suppose that's why I put that picture in there. And there's the rose. The rose is an interesting flower because we have done a lot of genetic altering of the rose to make more petals, In fact, some of the work we've done that have made them brighter and more beautiful and more petals on there have actually reduced the scent of the rose, which is too bad because the rose scent is very nice. And so some of our work has taken that away, so some of them don't smell as good as they used to. But apparently they're perfect flowers. They're all the stuff that's for the reproduction is down in the middle, though, covered up by lots and lots of petals. We've done that for the beautiful aspect of it. So an imperfect flower is how they're separated male and female. So here we have a male flower. You notice it's got just a narrow stem right there, but look at the female flower. What is that? That is the ovary, which is going to develop into a fruit. All right, so the female part is about making the fruit. You can see it cut open here. Now we see it cut in half here. You can see the male parts right there. And then cut in half here, you can see the ovary right there, okay? And so that's the female part right here. There are some of the more famous of the plants that are this way would be the pecan tree. So if you have a pecan tree and you've been mystified because it never makes any pecans, it might be because it is a male pecan tree. And it needs to have a female nearby, or a female pecan tree needs to have a male nearby so she can get the pollen from it, usually from wind and things like that. And then pecans can be made. Also, I'm going to show you a picture here in a minute of zucchini. Zucchini is also with male flowers and female flowers. So imperfect flowers. Here it is right here. All right, so here is the male flower. In fact, if you've grown zucchini before, zucchini is delicious, right? If you've grown it, you may have noticed a big flush of yellow flowers at the beginning, but if you look very closely, they're all male flowers. And so then you have to wait until the female flower finally gets sent out. It looks more like this. Here's the flower. Look what's on the end, a little tiny zucchinis there. Or I'm going to cut these open as well. Oops, didn't mean to do that. And you can see right here the male flower. Look, it's got the male part here with the pollen grains. We're going to see how those are developed here inside the filament and the anther. And on the female side, we have the stigma right here, which is going to accept the pollen and then come down in a very elaborate number of steps, and it'll end up making the fruit, like that zucchini right there. So this is back to our perfect flower. This is the one, if we were together, we'd be dissecting. And I'll talk to you about that when we get to our lab part. So the female part you're going to see here, it's perfect. So it's got the female part in the middle, the ovary, which is where seeds will develop. And then the male part is right here on the outside. On the tip there is where the anther is, and the anther is holding on to the pollen. You can see here it's called the microsporangium. The microsporangium make these little pollen grains, and we'll see how those form as well. It's actually quite elaborate. It's not simple at all what's going on with flowers, and we're still finding out new things about them. Notice that it's connected down here to the stem on the pedicel, and that we're going to see a peduncle here in a minute, and our sepal and our petals right here. So don't draw this. I'll show you what I think you should draw. In fact, here it is. Either you're drawing this in a blank box or you're labeling this that's already there. I can't remember. On my book, it's already there. So maybe you're going to draw the whole thing. So remember right here, you can pause the video so you can have as much time as you like to draw this in there. It's important you need to know these parts. We're going to start over here to the right, the carpel. We're looking at the female part of the flower right here. The old-fashioned word for it is pistil. Some people still use that word, but typically we'll see the word carpel now. So what does the carpel consist of? It consists of all the female parts of the flower. So here we go. Let's start with the big part here, the stigma. Sticking up high here is going to be available to accept the pollen onto it, okay? It comes down a long neck here called the style. It's like a tube right there. When you dissect, it's kind of nice because you can see how sometimes they're open right there with like a tube. Then we get down to the ovary, right down in here. And inside the ovary, we're going to have several little spots of the ovules. Guess what the ovules will become? They will become the seeds. Yes, the seeds. And then the whole thing's connected down here to the stem at the area called the peduncle. The sepal are these little like supportive leaves that are up there. They are green, so they're leaves. Not all flowers have that, but many of them do. All right, so there's the female part. The carpel includes the stigma, the style, the ovary, and ovule. Okay? Then over here is the male part. Okay, it's called the stamen. What is the stamen? It is these two parts. So the two parts of the stamen are the anther and the filament. The filament is holding the anther. It's holding it upwards to get it up and closer to the stigma over here. On the anther, it's going to have the pollen that gets created in a very elaborate way. All right, so like I said, go ahead and pause this, draw this out nice and neatly. You can color it different colors if you like. I sure hope someday I'm going to get to see your lab books again. So work on that. This is showing a photo of it, a little closer look right here. So we can see the female part sticking up, the stigma right here. Okay, it's interesting. It's sticking up past the male parts, right? So there's the style that's going to take it down to where it's going to end up at its ovary right there. We're just looking at the outer section here. And then we just see all the male parts, right? We see the stamen, number 3, and the filament holding the stamen up. Is that right? Yeah, and then we have the petal right down here. Okay, so let's go back for a second. Yeah, so I should have said that number 3 is the anther. The whole thing's the stamen, but it's really the anther, that part, and that's the filament. Okay, yeah, so I'll have to go back and change that. Okay, so there's lots and lots of kinds of flowers out there, and we're going to divide them up into a couple of different kinds, okay, about 3 different kinds. The first type is called the simple flowers. Simple flowers have one stem, one blossom, okay? So we're looking at a picture here of a tulip. It's got the one stem. It's interesting. I don't know if you can tell by looking here at that leaf that it has parallel veins, right? So if that's got parallel veins, you might know something about this flower already. It's a monocotyledon. So we're looking at a tulip, which is a monocot. We're going to see a video at the end here talking specifically about flower formation in monocots. They're actually some of the most beautiful flowers out there. This is the morning glory or buttercup right here. Apparently it's a single flower on a stem. That's why I put it there. It's harder to tell because you have the other flower right next to it. A rose, of course. Roses are famous for their long stemmed roses, right? And so one flower, one stem. You can see all those petals right in there. I was telling you about how they're all gathered around the middle part there. The next type of flower we have is called the flower cluster, where you have one stem with many flowers coming off of it. For instance, the bluebonnet that I showed you earlier. I'm not exactly sure what this flower is, but you can see all those individual flowers and then this one stem holding on to those individual flowers. So you may know what that is. I don't know. The composite flowers are amazing, okay? So these are the ones with a really big middle part right there. Think of a sunflower, for instance. So they have in the middle, that big round part in the middle. Each one of those little deals in there is a separate little tiny flower. Those are called the disc flowers. So the big middle part is called the disc. And around the edges, you have the ray flowers. So the ray actually has the flower plus this awesome thing that ends up being the petal, right? The petal of it. So that's the composite head. I'm going to show it to you in a photographic way here. So inside this disc right here, all of those flowers look like this. So here's the little flower. Here is the stigma style, right? And the stamens all right in there. The ones along the edge, however, have this special, here it's called the ligule right there. And then our style poking up right here. And these give the petals to the flower right there. Remember, those petals are going to be attractive to pollinators. They're going to have a scent to them that also attract. If you think about it, with a sunflower, if you've ever had an actual sunflower blossom that you look at, that big middle part right there, what we do is we let that dry out, and then each one of those flowers will have formed underneath here a sunflower seed, right? So you can pop all those little sunflower seeds out of one flower. So pretty cool. We're going to be looking at how pollen grains form now. Okay, so remember where the pollen is. The pollen is the male part of the plant. It is coming from the anther. So the anther is going to produce pollen grains. It's holding up on this filament here to hold it up high, and then there's these little germinal centers right here that are going to make a pollen sac and end up making a little. It's going to go through stages of cell division till it ends up with these microspores right here. And then forming this deal through the process of mitosis, a pollen grain. This is the male gametophyte. All right, so remember, this is part of sexual reproduction. We're going to see this cool deal. It's got this tube nucleus, which is amazing. It actually is a nucleus that digs a tube. Here it is showing the tube right here, because it's going to dig the tube all the way down through the style. And it also has this this generative cell right here. We're going to see this at the end. You'll see it in a video right there. All right, so there's our pollen grain. And it's going to take, you're going to see how this happens. It's going to end up digging down with the tube nucleus down toward the plant with 2 sperm cells. And those sperm cells are going to do something. Each one is going to have a specific job to do. All right, so think of those anthers as going through this process of making the little pollen grain, millions of them, so they can eventually dig down to where the ovary is. Here is a picture of, this is an electroscanning micrograph of different kinds of pollen. Out there, they're very cool looking. They look like little alien ships or something. You can see right here, there's a little crease on there. You'll see eventually in a video how that crease gets formed. And remember, that's one of the ways we tell between monocots and dicots is the number of creases they have on their pollen grains. All right, so pollen is all around us. Plants are releasing them all the time. Especially when they're in flower, obviously, because the flowers are the male part of the flower is going to have that pollen on there. Sometimes the pollen will be dispersed by the wind, and it'll just be in the wind. It'll get in our eyes and our nose, and possibly you have an allergy to pollens or different kinds of pollens. And that can cause an overreaction of your immune system, causing you to sneeze and feel pretty miserable and that kind of stuff. Well, that's what these little guys look like. Interestingly, we can look at ancient things and find from most of these things pollen grains, and we can make all sorts of interesting conclusions about what the people who made the cloth in the old days, something like that, what kind of plants they had. For instance, the mummies of Egypt, the mummies are wrapped with linen, and because the pollen could get caught in that linen while they were wrapping it, we can actually go back, take a look at some of that pollen, and figure out the kind of plants that the ancient Egyptians were growing. Yeah, so pretty fascinating stuff. The embryo sac formation goes through quite a process. So we're going to watch a little video that's going to help us understand it a little better.
Embryo sac formation begins with a diploid cell inside the ovule. That diploid cell undergoes meiosis to form 4 haploid cells. As is typical in female meiosis, 3 of those cells, called polar bodies, are small and quickly disintegrate. The one remaining cell then goes through an interesting form of mitosis where the nucleus is duplicated, but the cell doesn't divide. And in fact, that happens 2 more times, so that in the end we have a large cell with 8 haploid nuclei inside. At that point, cell division then occurs, but interestingly enough, it only forms 7 cells. The central cell is larger than the others, and it contains 2 haploid nuclei. This results in an embryo sac with 6 haploid cells and 1 large cell that contains 2 haploid nuclei.
Okay, so that is very strange, isn't it? Remember, this is the embryo sac formation. So this is happening down inside the female part, into the ovary, right? So this is the ovary of the plant. And we're going to see how the pollen grain of the plant, which would be the male part, is going to reach up, and it's actually going to come up into this little area right up in here. So why in the world is there all this elaborate cell division going on? We're going to see that each one of those different nuclei has a different job to do. And it's quite fascinating. Okay, in fact, you'll watch a longer video at the end. It'll give you a little more information on that. So pollination is going to take place now in that embryo sac, right? So let's watch a video on that one.
When pollen reaches the stigma of a carpel, its journey has just begun. The tube nucleus has to dig a pollen tube through the carpel so that it can get to the embryo sac, which is in the ovary. As the tube nucleus digs, the sperm cell follows. If there was only 1 sperm cell in the pollen grain, it goes through mitosis so that there are now 2 sperm cells. There have to be 2 sperm cells by the time the tube reaches the embryo sac because plants use double fertilization, which you'll learn about in a moment.
Okay, so our pollen has been made. We saw that elaborate thing, and now it's going to dig through with that one of the nuclei, right? The tube nucleus is going to dig down this deal here. And then it's got the 2 other sperm cells behind it. Okay, so that tube nucleus job is to dig into here and get in. And now we're going to see what happens with these 2 sperm. And they go through this deal, which happens in angiosperms, in the flowering plants, this thing called double fertilization, which is quite elaborate. Okay, so how does it happen? Over here we see what the last slide just told us, right? It's coming in like this. So then we see that the tube nucleus has done its job. So the first sperm cell will go into this area right here, okay, into this cell right here, our egg cell, and it will become a zygote. Look, 2N. That means it has all the genetic material it means. So that's the baby. That's the new plant right there. The second sperm cell will actually go up to this area right here where these 2 are in this large cell and fuse with these 2 polar nuclei up here. Okay, these cells up here are kind of almost like cushioning, and then down here we're going to see how they've done their job to receive the tube is what they've done right there. Okay, so what is happening here? We have a zygote, which will become our new plant, and then we have this thing with 3N, so triple, right? Triploid is what it's called. This one is going to become what's known as the endosperm. So think about a seed for a minute. In a seed, you have all that stuff in there besides the little plant. There's a little plant. We'll actually look at seeds next week. I'll have some pictures of seeds to show you next week. And then they have all that endosperm. Think of it like the yolk of an egg. It is going to be the nutrition to feed that zygote. So when I have a seed, it is dried out. And when I put it into the ground to plant it, I put water on it, right? That water all will go in there and it will activate the germination of this seed. And what will happen is it'll send down a root first, and then it will start to push up through the ground. Now keep in mind, the plant is going to make its own food through photosynthesis. It cannot do photosynthesis without sunlight. So when it's below the ground, is there any sunlight there? Nope. So it's going to need something to sustain it until it gets up to the place where the sunlight is. All right, so what is that sustains it? It's its yolk, right? It's its endosperm. Right there, the right word for it is endosperm. So this area right here is going to be enough nutrition to feed this until it can get to the sunlight and start making its own food. So cool, so elaborate. God is just showing off right here, y'all. It's not simple, the life that's going on inside flowers. Let's watch a little video about it.
The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the 2 sperm cells fertilizes the egg cell. This produces a diploid zygote, which will become the embryo. The other sperm cell moves up and fuses with both of the central nuclei, forming a triploid cell. This unusual triploid cell develops into an endosperm and serves as the embryo's food supply during early development. It is only angiosperms, flowering plants, which have this double fertilization characteristic, where a diploid zygote and a triploid endosperm form. Gymnosperms, pines, tracheophytes, ferns, and non-tracheophytes, mosses, lack this double fertilization feature.
Okay, so maybe that helped to explain it a little better to you so you can understand what's going on there. Now we're looking at pollinators now. How does the pollen get to the ovary? Okay, well we see how it digs through, but how does it get to the style at the top there? Well, you might think, well, it's right there next to it. Sure enough. So there are ways to get that pollen to get right over there to the female part of the flower. And when that happens, like for instance, wind can do that. You may have seen sometimes when the cedar trees are sending their pollen out. That usually happens in December. And these giant yellow clouds are all over the place. That's all pollen. So that is wind that's taking it to another cedar plant. Lots of people have allergies to that. But oftentimes, even perfect flowers like to get the pollen from another flower than its own. Okay, so it's the same type of flower, obviously, but it's going to get it from the one next to it instead of from its own anther right there. So pollinators are going to serve that purpose. Pollinators are very important. The most important, obviously, would be our honeybee. We've talked about the bees, right? We talked about these in our insect unit. But here's a picture of one. You can see that pollen all over him. Remember what the bee is doing. It's coming to the flower to drink the nectar and then taking the nectar back to the hive where it makes the honey to raise its babies, right? But it also uses some of that pollen to make the special royal jelly that it feeds to the larva that will become a queen. You can see that in this next picture with we call that the baskets, right? The bee basket right there. It's holding on to some of that pollen, packing it on there with some of the nectar. You can see that pollen all over this creature's fuzzy body, right? When he goes to the next flower, remember bees like the same kind of flower, she inadvertently will drop that pollen at the next flower, putting it onto the style and therefore going through the process of digging the tube and everything. Beautiful. We do need to take care of our bees. Bees are important to keep our plants pollinated. Even if we were able to pollinate our plants by hand, some gardeners do that, we could never get all the ones in the wild to do it. So it's super important that our bees remain healthy. Another pollinator would be our butterflies. You can see here the proboscis is extended down to get the nectar from the flower, and then the pollen would be collected upon its legs and things. Once again, an even body. They'll go to the next flower and drop off that pollen. You may have noticed hummingbirds out recently. They are also good pollinators. Notice that this one is going in, putting its long beak into this beautiful flower to get the nectar once again. And as it's doing, it's actually blowing around some of that pollen. It's picking up some of it on its feathers. Hummingbirds are really cool because they don't tend to land when they're feeding on flowers. They buzz around the whole time, and they're called hummingbirds because their wings are going so fast, right? They make a little bit of a hum. Also interesting, you may have noticed this, that hummingbirds are very highly attractive to red flowers. So if you have a hummingbird feeder, you may have noticed you put in some sugar water that's got red coloring in it, yes? One of the reasons I think God's designed them this way is because honeybees don't see red flowers. They don't see red. And therefore, they tend to not pollinate red flowers. So now we have these birds that come along that pollinate the red flowers for us. How about that? This brings us now to what our lab would have been this time, which would be our flower dissection. I typically try to get perfect flowers for you. Sometimes I can get big ones, typically not. I mean, mostly they're kind of small, so that's too bad. Right now, I did look for flowers and did not have any luck. I think flowers are not on the essential list or something. That was last week. Maybe this week they'll have some more flowers. But so what I did was I put in a little video here. You can see that with this perfect flower, we of course have our pretty petals. We have our style here. We have our anther, right, and our filament. Right here, you can see all that pollen right there. And over here, it's a different flower, but you can see it's been taken apart. And this part right here is basically what the dissection will be. Pollen grains, we would have taken some pollen grains and put them on a slide, looked at them under the slide to see what they look like from our flower. Not sure what these are from, but these are pollen grains from some plant. And you can see they kind of have those interesting little grooves on them. Remember, we have the grooves for the monocots and the dicots that are different that way. All right, this is a little video that I'll explain as we go through, as we're going to take our flower and dissect it. Okay, so we'll begin by taking the sepals off, just kind of peeling those off, and then we're going to do our he loves me not, right? Taking the petals off right there. And look what's exposed right there. That's all the reproductive parts. Here is the filament holding onto the anther, pulling off some of that, that pollen right there. We would put that under the scope. Now cutting through that, you can see that group right there of pollen tubes right there. That's pretty cool right there. I don't always see that. All right. And then we get down, of course, to where the action is. This is in the stigma down here. Cutting through that into the ovary. And pulling that apart. And we're going to cut that in half. And you can see into the ovary right there. This is why bigger flowers are better, because you have more to see right there. And right there in the ovary, you can kind of see the little dots right there. I'll show you a picture in a minute. Yeah, let me move on to the picture. So here you go. So we've cut through right here, and you can't, it's kind of shiny, right? But there's little circular things right here. Okay, and those are what are going to develop into the seeds. Very cool. So you can find that in the flower, the little pre-seeds, if you will. See, because when we cut the flower, we stopped its ability, right, to make seeds because we cut it. It's not growing anymore. That's because we enjoy using flowers for beauty, sometimes for the scent, this kind of thing. All right, so this video is a little bit long. It's going to extend your lecture a little bit because the lecture was a little short, but it's going to go back over all these things we talked about. There's way more information in here than you need, but it's all accurate and it's very well done, in my opinion. So just sit back and relax. Take a look.
For most of us, flowers are a source of pleasure, with their variety of shapes and vast range of colours. They're used to brighten our homes and gardens. And their bright blooms also bring colour to our city streets. But flowers are also the reproductive organs of the plant. It's for this reason that scientists work with flowers to breed new, better and more valuable varieties of plants. A flower is therefore not just decorative, but plays an important part in plant reproduction. The role the flower plays in reproduction is easier to see in the larger, simpler flowers of the many monocotyledons. The small number and large size of their flower parts allows us to easily illustrate their structure. Each flower has 3 sepals, which overlap 3 petals. Inside the petals there are 6 stamens that produce pollen. Each stamen is attached to the flower by a filament. The enlarged sac at the tip of the filament is called the anther. Inside the anther there are 4 microsporangia. Each microsporangium is nourished by a layer of surrounding cells called the tapetum. A microsporangium consists of diploid sporocyte cells. The cellulose walls of these cells are soon replaced by a layer of callose. Each sporocyte now divides twice by meiosis. This creates 4 haploid microspore cells, each with 1 set of chromosomes. This group of adhering microspores is known as a tetrad. Soon the callose is removed and sporopollenin begins to form on the surface of each cell. The surrounding tapetum now releases additional sporopollenin. One area of each wall, the sulcus, remains smooth, receiving very little sporopollenin. However, the rest of the wall is covered in a thick, ornate coating of this tough, resistant material. Each microspore nucleus now divides by mitosis. The lower generative cell separates from the wall and moves into the cytoplasm of the larger tube cell. This is the immature male and is known as a pollen grain. Before it is released, the pollen grain accumulates nutrient reserves. Finally, most of the water is lost, and the pollen grain enters into a resting state. Before the anther releases the pollen, the cells beneath the epidermis develop thickened walls. This is called the endothecium. When the pollen and the anther mature and dry out, the endothecium shrinks. This tears open the anther between each pair of microsporangia, allowing the pollen to escape. At the center of the flower, there are 3 carpels. At the base of each carpel, there is an enlarged ovary. A slender style extends from the ovary. And at the tip of the style, there is a stigma. The 3 carpels are fused together, forming one stigma and a style with a central canal. The base of the canal leads into each of the 3 ovaries. In the young carpels, small outgrowths appear on the ovary wall. Each of these is a megasporangium. 2 layers of cells, the integuments, grow over the megasporangium. A small pore, the micropyle, is the only area that remains uncovered by the integuments. This entire structure is called an ovule. Inside the sporangium, there is a single diploid sporocyte cell. The diploid sporocyte divides by meiosis to form 4 haploid nuclei, each with one set of chromosomes. In some plants, such as Lilium, walls are not formed around these nuclei. But in the majority of flowering plants, walls are formed around each nucleus, resulting in 4 megaspore cells. 3 of these then degenerate. The surviving spore enlarges, and the nucleus divides 3 times, yielding 8 haploid nuclei. 2 of these nuclei move towards the center. These are known as the polar nuclei. 7 cells now form. At the micropylar end, there is an egg cell and 2 synagid cells. In the middle, there is a central cell containing the 2 polar nuclei. Above this, the remaining nuclei form 3 antipodal cells. This is the female gametophyte, also known as the embryo sac. This is contained within the megasporangium, which is covered by the integuments of the ovule. The ovule, in turn, is surrounded by the carpel. The transfer of pollen to the receptor surface of the stigma is called pollination. This happens by various means, such as wind, insects, or other animals. The pollen grain now absorbs water and begins to swell. A pollen tube soon emerges containing the tube nucleus and the generative cell. The generative cell divides to form 2 sperm cells. In order to reach the ovaries, the pollen tube must now carry the sperm down the full length of the canal. It does this by first growing through the stigma to reach the surface of the canal directly below. Here it grows along the moist transmitting tissue, which serves as a path to the ovary. Pollen tubes have a very fast rate of growth, often several micrometers per minute. This growth is localized at its tip. Vesicles containing wall material accumulate at the tip, where they are required for wall growth. Behind this, the cytoplasm flows around the cell in regular streams. This is known as cytoplasmic streaming. The 2 sperm cells and tube nucleus continually move forward as the cell grows. Plugs of wall material called callos form, which keep the cytoplasm close to the growing tip. When the pollen tube reaches the base of the style, it grows into one of the 3 ovaries. Inside the ovary, the tube locates the 2 rows of ovules. Approaching an ovule, the pollen tube grows into the micropyle, where it penetrates the megasporangium. It then grows into one of the synagid cells, releasing the 2 sperm. One sperm fertilizes the egg cell, forming a diploid zygote cell. The second sperm fuses with the 2 polar nuclei, forming a primary endosperm cell. This double fertilization is characteristic of all angiosperms. The ovary contains hundreds of ovules, and each requires a pollen tube to be fertilized. To accomplish this, the style supports the growth of a large number of pollen tubes. These grow down the style and enter each of the 3 ovaries. When the pollen tubes enter an ovary, they each locate and fertilize an ovule. Each ovary now has many fertilized ovules, which will develop into seeds. After the ovules are fertilized, the flower undergoes several changes. Many parts begin to wither. Eventually, these parts are shed. Only the ovary remains healthy and firmly attached to the stem. Inside the ovary, the ovules are now about to undergo many changes. Each ovule contains 2 fertilized cells. The nucleus of the primary endosperm cell begins to divide to form endosperm tissue with no cell walls. The zygote divides, forming a large basal cell and a small apical cell. The basal cell produces a chain of cells called a suspensor, and the apical cell gives rise to an embryo. The suspensor now begins to transport nutrients into the young embryo. In monocotyledons, a single seed leaf or cotyledon forms. As it enlarges, the embryo also receives nutrients directly from the surrounding endosperm. During this time, walls slowly form within the endosperm tissue. A tiny shoot tip forms on one side of the embryo. Below this, there is a stem called the hypocotyl. At the base of the hypocotyl, there is a root. This is covered by a protective root cap. In most monocotyledons, nutrients now begin to accumulate within the endosperm. When nutrient accumulation is complete, much of the water is removed, and the embryo enters a state of dormancy. However, a different sequence of events takes place in the ovules of most dicotyledons. In these ovules, the growing seed leaves accumulate nutrients and absorb all of the endosperm. As both types of ovules mature, the integuments develop into a tough, protective seed coat. The ovule is now a seed. At the same time that the ovule is changing into a seed, the developing embryo is releasing hormones. These stimulate the ovary to expand and make room for the enlarging ovules. In this way, the ovary wall expands and is transformed into a fruit. In most lilies, the fruit is a tough, dry capsule that tears open to release the seeds. The seeds are soon dispersed and will germinate to form new plants, thus completing the life cycle. We have seen that flowers capture pollen and ensure the success of fertilization and seed formation. In the plant world, reproduction by flowers is the most efficient. It's therefore not surprising that a great variety of flowering plants dominate the landscape. Many of these attain great size, but others are tiny and almost microscopic. Some flourish in places where water is scarce. Others thrive in ponds and lakes. And from this vast variety, we have, by careful breeding, developed all those plants which provide us with food. With the great variety of flowering plants, the world in which we live is indeed a very colorful place.
Okay, I hope you enjoyed that. If you stuck with me the whole time there. So our flower dissection was our lab, and looking at pollen under the scope. We would have looked at that today. Maybe looked at different types of pollen. I've started a germination bag for next week. You can do this at home if you like. You can take some seeds, any seeds you might have that are extra, even beans, dried beans you may have in the kitchen, and put them into a Ziploc bag with a wet paper towel. And then next week we're going to look at those. They start to germinate. We'll see that little root coming out. Okay, gang, so that's it for today. We're at the end of our lecture, so enjoy it, and I will see you soon, I really hope. All right, bye.